Welcome back. So this is the pre-lab lecture for RT-PCR3. In this week's lab, we are going to be determining the concentration as well as the purity/integrity of our RNA samples that we've isolated in either the week previous or previously in lab. So just an overview of this week's lab. The primary goal and objective of today is again to determine the concentration and purity/integrity of our RNA samples. And we'll be doing this in two ways. Just as a reminder, this is RT-PCR3, so this is our third activity that we're doing through section 1, or RT-PCR. So the first thing you're going to want to do is thaw our samples on ice. It's going to be critical to keep all of our samples on ice today anytime we aren't using them, right? So if you remember, RNA is very sensitive and prone to degradation. So we're going to want to protect our samples and do everything we can to maintain that RNA integrity. So the first thing we're going to be doing after our samples are thawed is using the nanodrop to calculate the concentration of our RNA, as well as take a general reading to see what the ratio of protein to RNA is in our samples, right? See how well we did last week or in previous weeks separating out our RNA from our protein. After we have that, we're going to do a quick gel electrophoresis experiment to determine the quality/purity of our RNA using a MOPS buffer. The MOPS agarose buffer produced gel. And lastly, we're going to do some calculations to calculate the concentration of our samples and then how much volume of our samples we need to prepare our reverse transcriptase reactions that more than likely your instructor will do for you, but it'll be important for you to do the calculations yourself. So as a reminder from our RT-PCR overview video, right? So last week or previous weeks, we isolated all the RNA and DNA from our samples. So recall that this is mRNA, tRNA, ribosomal RNA, and various other forms of RNA. And ideally, we've excluded all of our genomic DNA, but it's possible that we have a little bit left. And so what today we're doing is calculating how much RNA we need to prepare and provide for our our RT reactions, right? Our reverse transcriptase reactions. See how much of our RNA samples we need to provide and set up so that we can actually perform our reverse transcriptase reaction, which is going to selectively convert our mRNA, right, using that poly A tail and our oligo DT primers, to selectively convert all of our mRNA, into cDNA. So this is the calculatory steps that are involved in this process, and that's what we're doing this week. So over here on our right is an image of a nanodrop. Ours looks very similar to this. It doesn't have this wire hanging out. The wire is encased in a small plastic covering, but it works the same way, right? So at the beginning of our lab, we can essentially have our groups break up into two sections. If you have volunteered or you get chosen to pour the MOPS gels for your group or the lab, we'll be combining multiple groups on one gel. Make sure you work with our TAs or your instructor to properly measure and pour our agarose MOPS gels. And we'll be doing that over by the microwave right around table 5. So if you're not doing that, you and your group members can come over to the nanodrop, which is in the back part of the room, close to the 96 well plate that we've possibly used before. And you will work with either our TAs or your instructor to take several measurements, right? So the nanodrop is basically a very small spectrophotometer that's going to measure the absorption of our samples at 280 nanometers as well as 260 nanometers. So when we do this, your instructor or your TA will give you a quick run through. We'll also blank the machine for you, but there's essentially a little probe right under where my laser pointer is pointing. We'll open this arm, place our samples on that probe, close this arm, and then take our readings. And 280 and 260 are important because 280, or the absorption of our samples at 280, will tell us how much protein is in our sample, and then the absorption at 260 will tell us how much DNA/RNA is in our sample. And this will also give us a 280 to 260 ratio, which our protocol explains where we want those numbers to be. Right. This will essentially tell us what the ratio of protein to nucleotides in our samples are. Once we have, and again, in addition to that, we will also get the concentration of RNA in our samples. Once we have the concentration of RNA in our samples, we will use that concentration to calculate the volume of our sample that we need so that we equally load our reverse transcriptase reactions. So a little bit more about the nanodrop, right? I mentioned this previously, just in the previous slide. So our nanodrop is going to take two absorbance readings, one at 260, where again the essentially nucleotides absorb wavelengths of 260 due to the double bonds and pi electron structures in their ring formations. And then we'll also record, and the information will be given to us, the absorption at 280 nanometers. And this is partially a quick and dirty way to calculate protein concentrations, because amino acids such as tryptophan and tyrosine absorb wavelengths at this light. And to a slightly, I wouldn't even say slightly lesser extent, virtually none, phenylalanine will also, right? And the reason these amino acids selectively absorb light is, again, due to that ring structure and the conjugated pi electron bonding and components of these bonds within the amino acids. Again, this is a relatively quick and slightly less accurate way to calculate protein concentration than we will use in other sections of the lab. Up here in the top right is basically an absorbance spectra. Hopefully everyone is familiar with looking at these from your experiments and lab experiences in OChem. So we will essentially get this absorption spectra. It'll look different for every sample. We will have our nanodrop software set to RNA, and then ultimately what we want is the concentration of our RNA. And again, that'll be spit out in units for concentration. So this would mean nanograms RNA per microliter sample, right? And pay careful attention to the units that come out of our readings, right? The units that come out of our readings might not directly match the units that we need to calculate our samples in. So be aware of units and be aware of unit conversions. And I meant to load these in earlier, but again, our nucleotides such as A and G, adenine, guanine, and thymine and cytosine will absorb light at 260, and then our the ringed R groups on our amino acids such as tryptophan and tyrosine will absorb light at 280. Now you might be asking yourself why it's so important to calculate the concentration of RNA in our samples, right? So over here, you can think of these maybe as our control, drug 1 and drug 2 samples from our experiments in lab. And as you can see here, right, we're going to assume that all of these, we're going to assume that each of these samples has a different concentration of RNA present in each sample, right? So over here in our control, we have a concentration representative of this many number RNA molecules. Our drug 1 has a lower concentration, right? There are fewer RNA molecules. And then our drug 2 has a very, very high concentration of RNA, right? So if we were to take the same volume from each of these samples, right? Say we take 1 microliter from each sample, the total number of RNA molecules in that 1 microliter of sample will look different, right? So in a much more concentrated sample, such as our drug 2, 1 microliter of sample will contain many, many more RNA molecules than, say, our drug 1 replicate that has a lower concentration, right? Less nanograms RNA per microliter than drug 2. And then our control is over here. So why this is important for keeping in mind during our reverse transcriptase process, if we were to load that equal volume into our reverse transcriptase reactions, right? Remember, reverse transcriptase is going to convert every mRNA in a one-to-one fashion to cDNA, right? So let's perform our reverse transcriptase reaction, and now we have just as many cDNA molecules as we had mRNA molecules. So what we've done here is in our control, we've pretty much produced 1, 2, 3, 4, 5, 6, 7, 8 cDNA molecules. In our drug 1, we produced 4, and over in drug 2, we produced many, many, many. Right? So if we were to continue on the process and load these cDNA samples into our wells in section 5, or excuse me, in RT-PCR 5, when we actually run our DNA gels, we could run our DNA gels and see something like this, right? So we would have a medium band in control, a very light band in D1, and a very, very bright band in D2. And what you may want to do is assume that this means in D2 that our drug has elevated the expression of our gene of interest, right? That may be what you want to assume because it has a brighter band. But in reality, this is technically a false positive because we overloaded drug 2 with many, many more copies many more copies in total of cDNA, right? There's literally a larger quantity of cDNA loaded into this sample. So naturally, you would expect it to be brighter. Whereas over here in D1, we loaded fewer total cDNA or fewer total mRNA into our reactions, and thus we have a much lighter band, right? So these would be considered incorrect results because we incorrectly primed our reactions. So in this example, we are going to normalize the total quantity of RNA that we load into our reverse transcriptase reactions so that ultimately when we run our DNA gel in part 5, the intensity of the bands directly reflect the actual concentration or expression levels of our gene, right? So as you can see in our samples, we still have different concentrations of RNA in our samples, right? Similar to the previous slide. Our lowest concentration sample over here is D1, our highest concentration is D2, and our middle concentration over here in control, right? But what you will notice now is that there are different ratios of our gene of interest in each of these samples, right? Over here, this looks to be about 3 copies of mRNA of our gene of interest to 5 copies of other mRNAs, whereas this is 1 to 3, and then however many this is, I'm not going to count it. But you can visually see that there are more copies of our mRNA of interest compared to other mRNAs, right? So if we normalize the quantity of mRNA that we send into our reverse transcriptase reactions by calculating concentration and taking a specific volume that will transfer only 5 mRNA molecules, now in our reverse transcriptase reactions, we have a total of 5 mRNA molecules in each sample. So when we perform our reverse transcriptase, again, we produce a 1-to-1 copy of mRNA to cDNA. So now we have in total, we have 2 copies of our gene of interest and then 3 copies of other genes, right? So this will be starting material that we send into the PCR section in section 4 of RT-PCR. Whereas in drug 1, we only have 1 copy to start with, and in drug 2, we have 3 copies to start with. Right? So after our PCR, our polymerase chain reaction, these copies hopefully will selectively increase, but remember, in every cycle of polymerase chain reaction, our gene of interest will double, right? So this guy, control, is going to double at a faster rate. It'll double at a faster rate than our drug 1. whereas our drug 2 will double the fastest because we are starting with more copies of our gene of interest. So now, when we run our DNA gel, we can actually believe and interpret that a brighter band in our DNA gel means that our drug has increased the overall expression of our gene of interest, right? And that's why it's important to make sure you're loading the same quantity of RNA into our reverse transcriptase reactions so that we don't unintentionally get a false positive that was just due to overloading a sample with more mRNA to start. We want to start with the same quantity of mRNA in our reactions. And more immediately, this week in lab, we are still going to calculate concentrations of our samples, and when we load our RNA gels, we want to load the same quantity of RNA into each well. This is going to help us make sure that our bands are equally loaded, so when we interpret our results, any differences between groups isn't a result of misloading, it's just a result of the integrity of our samples. So once we've done all this, we're going to load our RNA gels with the same quantity of RNA. We are going to flip the power on, and again, this is a quick and dirty visualization of our RNA quality. And what we're hoping to see is two bright bands that represent our ribosomal RNA. So when we flip the power on, remember that our RNA is primarily negative, so it could be considered an anion, and anions run toward the anode, right? So it's going to run towards the positive side. And once we turn the power on, what we're hoping to see is over here, right? In lanes 3 and 4, we want to see two separated out bands. And this will be, if you've read the protocol, this will represent the ribosomal 28S and 18S ribosomal RNA bands. In a sample that's degraded, we will see this smearing of RNA, right? This is undesirable, and we don't want to see this. So, just a few last minute notes. We're going to be using, again, MOPS running buffer for this. We will have between 8 and 15 lanes per comb. Our TAs or your instructor will help load the RNA ladder. So we'll essentially be doing that for you. The information about the RiboRuler high-range RNA ladder is loaded up on Brightspace, so make sure you check out that. You will need the information from the band size contained in the manufacturer information uploaded to Brightspace again. Make sure you're loading the correct volume for your samples, and if you're interested, we are running these gels at 120 volts for 30 to 45 minutes. In addition to your instructor keeping an eye on how your gels progress, make sure you are also keeping an eye on how that diaphragm moves through our gel. And as always, remember, you can always email me ahead of time with questions, or swing by office hours, or ask before lab starts, or you can even ask during lab. See you this week.